Our speaker is Adam Searles. Adam is a PhD candidate. For those that you don't know, when you pass your qualifying exam, you become a PhD candidate. You used to be a student, and Adam just passed his qualifying exam last week. So he's now a candidate. He's in the School of Natural Resources and Environment and is also part of the Nature Coast Biological Station. Here at UF, he is supported by a fellowship from the National Science Foundation. He's also supported by Florida Sea Grant through a Guy Harvey Fellowship. Before coming to UF, he was a student at the University of Central Florida, and he had many recognitions there as well. In fact, he was just recently named 30 under 30 for UCF. In between his bachelor's and his master's, Adam also worked for FWC, so he has some practical experience as well. His research interest I put down as fisheries and habitat linkages and coastal community ecology in Florida. And that Florida, I think, is important. Adam is a native Floridian, and a lot of the stuff that the questions that he asks are based on things that natural history that he's seen in Florida or issues that are rising locally, and he really addresses questions that are applicable. In addition to his research, he's also really does a really good job mentoring undergraduate students, and I think that's a real priority for Adam. He runs a program to mentor students, undergraduate students in environmental science and in marine science who are interested in applying to national fellowships. And he himself was a Hollings Scholar, which is a NOAA fellowship as an undergrad, and that's one of their highest ranked fellowships. And he, in the past couple of years, has helped 4 undergraduates get those fellowships. Adam's a great member of our team, and I'm excited for you to hear his research. And with that, I will let him take it over.
Well, thank you, Laura, for the very kind introduction. All right. Can everyone see the proper screen?
Yep.
All right, here we go. Hi, everyone. My name is Adam Searles. Like Laura said, I'm a PhD student here at UF. And today I'm going to be talking about submerged aquatic vegetated habitats in the coastal regions of Florida. And the specific title of my talk is Seagrasses versus Macroalgae. How do animal communities differ across habitats? And before we talk about animal communities, I want to talk specifically about the organisms that constitute these habitats. The first of which is seagrass. Seagrasses are plants. They have a lot of things in common with terrestrial plants, especially terrestrial grasses. They have complex below ground root structures. They have individual shoots that shoot up into the water column, and they have multiple leaves per shoot that create complex, large meadows, just like terrestrial grasses do in grasslands or prairies. Next is macroalgae, and as we can see here, macroalgae have a very diverse set of morphologies. We see reds, greens, browns, attached, drift algae, etc. Both of these groups, seagrasses and macroalgae, can be defined as macrophytes, and all the macrophyte is a macroscopic photosynthetic organism. So throughout the rest of the talk, when I say macrophyte, I'm referring to seagrass and macroalgae. And so you might be wondering, well, some of these things kind of look similar between seagrasses and certain macroalgae. What determines what's a seagrass and what's a macroalgae? And really, it's the evolutionary history of these two groups that defines each one as a macroalgae or seagrass. So here in this figure, we have geological time across the x-axis, if you will, here. And over on this side, we have farther back in evolutionary time. And what we see here is green and red macroalgae emerging approximately over 400 million years ago. They exist only in the marine and aquatic environments, and they've existed there for a long time. They've speciated, adapted, and evolved for over 400 million years in the marine and aquatic environments. And as we move forward in time, we saw that one ambitious macrophyte emerged onto the land a little over 200 million years ago, and from there speciated into many, many, many species that we call the plants. And so from there, about 100 million years ago, one even more ambitious plant adapted back into life in the marine environment. And from there, speciated into approximately 60 species that we call the seagrasses. So seagrasses are part of this closely related group of plants that live in the marine environment, and they're all descended from one common ancestor. Whereas red and green and brown macroalgae, for that matter, have always existed in the marine environment. They're a relatively basal group. They're not super complex in terms of their morphology, but they do have a wide diversity of phenotypes because they've been evolving in that environment for so long. Whereas seagrasses, on the other hand, as I said before, are relatively complex, but don't have quite as diverse a set of morphologies. Now, I've spent a minute talking about this. I don't want to get too hung up on the differences. Just know that there are evolutionary-based differences between seagrass and macroalgae. Now, that said, both of these groups are very important for coastal ecosystem processes, and that's why I don't want to get too hung up on evolutionary differences. Both macroalgae and seagrass are really important for carbon storage, nitrogen cycling, foraging grounds, nursery habitat, protection from predators for smaller organisms, and much, much more. And I think it's important to say that because macroalgae these days seem to get a bad rep from news and media outlets, especially here in Florida, because of a lot of the coastal anthropogenic degradation going on in Florida in systems like the Indian River Lagoon or Charlotte Harbor. We see lots of seagrass die-offs and subsequent increases in macroalgae abundance, and macroalgae tend to get the blame for that. However, they're merely responding to favorable conditions, and in healthy coastal ecosystems, both seagrass and macroalgae serve very important roles side by side to generate the ecosystem services and help coastal ecosystems function properly. Now, that said, some of the local examples that we'll be talking about today of seagrasses and macroalgae are, for the seagrasses, turtle grass, Thalassia testudinum, in other words, and shoalgrass, also known as Haloduliridae in scientific nomenclature. Looking at the macroalgae, we'll be talking mostly about Caulerpa paspaloides, this palm tree-looking species of macroalgae, and Caulerpa prolifera, this flat-bladed curly one. I'm not aware of any really popular, well-known common names for either of these macroalgae species. So if anyone knows, please let me know. And because of this, I'll be referring to all of these macrophytes via their scientific name throughout the rest of the presentation. And so it's the differences between these two things in terms of ecological function that I'm really interested in. We know there's differences between seagrass and Caulerpa in terms of the things they do in coastal ecosystems, but it's really hard to determine what those differences are. One of the most general statements I can make is that differences are present between seagrass and macroalgae across space, time, and species, and they vary a lot across these different scales. So for example, when we talk about sediment trapping is the ability of submerged aquatic vegetation to slow hydrodynamic forces and allow particles that are suspended in the water column to settle onto the bottom. This increases water clarity, which is really important for these species to grow and thrive and create complex beds. So water clarity is a good thing. So there's actually a lot of interest in the sediment trapping ability of these two different groups. And several studies in the Mediterranean that have looked at sediment trapping between macroalgae and seagrass have found that in the summertime, macroalgae have improved sediment trapping capabilities compared to local seagrasses. However, in the winter, they have very comparable sediment trapping rates. So we see variability over time. Moving just outside of the Mediterranean to the Canary Islands, this study, Toya et al. 2014, found that seagrasses always have an improved sediment trapping capability compared to local macroalgae. So we see variability across space as well as across species of macroalgae and seagrass. So you can see why it's really hard to define which one is better at a certain thing because of how much variability there is. Now, one of the main differences in ecological function I'm interested in is habitat provision. Several studies have found that seagrasses tend to support relatively complex trophic structures with a diverse set of fishes and many trophic levels, whereas Caulerpas tend to support a condensed trophic structure with a high diversity and abundance of small invertebrates and the fish species being invertivores, things that eat these invertebrates as the top predator in the system. Now, for us as humans, trophic complexity tends to be a good thing because the more trophic levels we have, the higher level predators we have, and it's those higher level predators that we're interested in for both recreational and commercial fishing opportunities. So they're really important, one, for fun, and two, for coastal economies. That's the word. And it's this difference that we see between seagrasses and Caulerpa that has really become the major motivation for my dissertation, because we know that there's differences, but we don't really know why there's differences. And so this chapter of my dissertation is focused on quantifying those community level differences between seagrass and Caulerpa habitats in a relatively pristine estuary. And I want to take a second to talk about those last two words, pristine estuary. Like I said before, macroalgae kind of has a bad rap because of, we see anthropogenic degradation in estuaries, and subsequently we see macroalgae taking over. However, in pristine estuaries, again, seagrasses and macroalgae work side by side to support the animals that constitute the food webs of our coastal ecosystems, and much, much more. And so for this study, we needed a relatively pristine estuary that had a natural abundance of both seagrass and macroalgae. And we found that estuary in Crystal Bay, Florida. Crystal Bay is here in the Nature Coast. It's relatively free of impacts from eutrophication, habitat degradation, etc., and also has documented lush seagrass and macroalgae beds all the way back to the 1950s. And so this served as the perfect place to conduct our study for understanding how seagrass and macroalgae contribute to ecosystem function in pristine natural settings. And so in Crystal Bay, we designated 11 sites here, ranging from 100% Caulerpa cover to 100% seagrass cover. As a reminder, our main Caulerpa species are Caulerpa paspaloides and Caulerpa prolifera for our macroalgae. And then the main seagrass we're focusing on for this project is Thalassia testudinum. So at each of these 11 sites, we used a plant corer to measure the above ground biomass of each plant species in a standardized area. And then we paired these data with fauna data from our benthic sled. Now, a benthic sled is a relatively large contraption that you pull through the water, and it skirts over the top of seagrass and macroalgae, and it scoops up all the small invertebrates and fishes that use these habitats. And by relating these two data sets, we're able to see how the fauna respond to the macrophyte abundance at those sites. And so when we take a preliminary look at these data, we see here this is proportion of SAV biomass on the y-axis across our sites on the x-axis. Each color represents a different species of macrophyte, with blues generally being macroalgae and green generally being seagrass. And so dark blue in this case is Caulerpa paspaloides, light blue is Caulerpa prolifera, and green is Thalassia. So when we look at sites 1 and 2, we see that these early sites are dominated by Caulerpa paspaloides. And when we move to our sites 6 through 9, we see a greater mixture of macroalgae and seagrass in Caulerpa prolifera and Thalassia. So we call these our mixed sites. And then when we move over to sites 10 and 11, we see that these sites are dominated by Thalassia, high seagrass biomass. However, when we look at the spring time period, we sampled all these sites in fall and spring, we see a much more homogeneous kind of random mixture of all three of these macrophytes across all of our sites. And when we take a more statistically rooted look at these data using a principal component analysis, we see that exactly what we'd expect based on these data. So this is a PCA. It's an ordination method, which is just a fancy name for this type of analysis. And all you really need to know is that each point here represents one seagrass core or one plant core. And the closer 2 points are together, the more similar the macrophyte community is in those samples. And the farther apart they are, the more different the macrophytes are between those two samples. And so when we look at the PCA, it's telling us as we move this direction in ordination space, we have increasing Thalassia biomass. So when we look at these samples here that are clustered together, we see that they come from sites 10 and 11 by these labels. And so when we look over here at sites 10 and 11, we see they're dominated by Thalassia biomass. So the PCA is telling us exactly what we'd expect. When we look over here, we see that this is the Caulerpa paspaloides dominated samples. And we also see that these samples come mostly from sites 1 and 2. Again, exactly what we'd expect. It's dominated by Caulerpa paspaloides biomass. And finally, when we look at our last cluster, we can call this our mixed sites. We see that they're characterized by Caulerpa prolifera biomass, as well as Thalassia biomass, and that they all come from sites 6 through 9. Again, exactly what we'd expect. So we see these differences in our graph here clearly in statistical space. Now, when we look at our spring data, again, we had a much more homogeneous mixture of plants across our sites. And then when we look at the macrophyte PCA here, we see that homogeneous mixture of macrophytes across all of our sites. Not really any clear patterns. It's really one homogeneous continuum of macroalgae and seagrass mixture. And so moving on to our fauna data, these are the benthic sled data that we collected at each of these sites. This is a non-metric multidimensional scaling, which again is another ordination method. And all you need to know is that each point is one benthic sled sample, and the closer 2 points are to each other, the more similar the animal communities in those benthic sleds. The farther apart, the more different the animal communities. And so on this ordination, we have both fall samples in the circles and spring in the triangles. And the colors correspond to the habitat type that those samples were collected in. So here we have in the blue, Caulerpa paspaloides dominated, red is our mixed sites, and green is our seagrass dominated sites. So taking a look at the fall data, we see that the animal communities cluster really close together based on which habitat they were collected in. So here we have our Caulerpa paspaloides, here we have our mixed sites, and here we have our seagrass dominated sites. So we see high similarity in the animal communities based on which habitat they were collected in. Now, when we move down the y-axis towards the spring samples, we see that spring is separated from fall, meaning that the animal communities are different across time. But also we see kind of a homogeneous mixture of the faunal community, very high similarity in terms of the animal community living in these grasses across all of our sites during the spring. And when we take these data and compare them to our macrophyte data, so here is the fall PCA for our macrophytes, and here's the fall faunal data. So we see exactly what we expect based on our macrophyte data in the fauna data. We see high clustering, high differences in the dominant macrophyte type at each of these sites. We see distinct animal communities according to what habitat types are present at those sites. However, when we look at the spring, when we had a homogeneous mixture of macrophytes across all of our sites, we see a very highly similar homogeneous animal community. And so based on these analyses, we can say that one, seagrass and macroalgae do support unique animal communities. We saw that in the fall when we had unique habitats. We saw unique animal communities. And we can also conclude that these animal communities reflect macrophyte abundance and identity. Again, we saw that in the fall. And finally, we expect animal communities to change when relative abundances of seagrass and macroalgae change. And so I think it's really interesting to take this understanding of animal use of seagrass and macroalgae in pristine conditions and compare them to places where we don't have pristine conditions, where we have much more anthropogenically influenced regions. And we can use this data as a baseline, understanding that there are faunal differences between seagrass and macroalgae, and that animals respond to changes in the abundance of seagrass and macroalgae, and see how these differences interact with things like eutrophication, climate change, and other multiple stressors. So for example, this project takes place in the Indian River Lagoon, which is a subtropical bar-built estuary on Florida's east coast. And for some background for those not familiar with the Indian River Lagoon, over the past 15 to 20 years, we've witnessed major declines in the abundance of seagrass in the Indian River Lagoon. And so some people actually estimate we've lost up to 95% of seagrass cover in the northern Indian River Lagoon in this time period. Now, as a result of that, Caulerpa is actually now the dominant macrophyte in the Indian River Lagoon. Furthermore, the Indian River Lagoon is also experiencing firsthand the effects of climate change, and specifically tropicalization. Tropicalization is the phenomenon whereby tropical organisms move towards the poles as water temperatures increase in those regions, and they're now suitable for life for tropical organisms. And these figures specifically come from a study that I was a part of at FWC before I came here to UF. And they show the rise and changes in distribution of the Western Atlantic sea bream, which is a tropical herbivore. So specifically, the figure on the left shows a hotspot analysis, which means that the red colors are significantly high areas of sea bream catch, and the blue areas are areas of significantly low sea bream catch. And so we see here in the left panel that in the years 1998 through 2003, we had very low catches of sea bream throughout the Northern Indian River Lagoon, save this one net here that had a few in there. However, when we do that analysis on the 2015 through 2019 time period, we see a lot more significant hotspots of sea bream catch in the Northern Indian River Lagoon, specifically around the Sebastian Inlet area. Now, this wasn't just a few nets that had a few species. This poleward shift in the, or excuse me, poleward expansion in the distribution of sea bream was affiliated with a almost 49-fold increase in the abundance of the Western Atlantic sea bream in this area. And that's shown here in the blue line, which from now on, blue will always represent the tropical sea bream. At the same time, we witnessed major declines in the abundance of local sheepshead. And this is concerning for a few reasons. One, sheepshead are a very important local fishery species. And two, sheepshead in the Northern Indian River Lagoon are actually herbivorous. Throughout most of sheepshead range, they consume things like small crabs, barnacles, invertebrates, etc. However, in the Northern Indian River Lagoon, they have a unique jaw morphology, which better suits them to consuming seagrasses and macroalgae, etc. So unfortunately, it's this uniqueness that sets them up perfectly for competition with the newly arrived tropical herbivore, the Western Atlantic sea bream. So the reason this is even more so concerning is that as sea bream move poleward, they have the potential to graze on already extremely imperiled seagrass populations characteristic of the Northern Indian River Lagoon, but also compete with dietary resources with sheepshead. And this could actually be a double whammy for sheepshead because sheepshead also rely on seagrass for nursery habitat. So sea bream serve this compounding threat for sheepshead in the Northern Indian River Lagoon. And it's based on these concerns that we wanted to know, do sea bream consume seagrass in the Indian River Lagoon? Do sea bream diets overlap with sheepshead? And how do seagrass and Caulerpa availability affect these diets? So to answer these questions, we worked with FWC's Fisheries Independent Monitoring Program in Melbourne, Florida, to collect sea bream and sheepshead from this area of the Indian River Lagoon here on the left, because that's where we saw the greatest increase in sea bream abundance. So with FWC's Fisheries Independence Monitoring Program, we collected 93 sea bream and 66 sheepshead. We brought these all back to the lab for gut content analysis, where I excised their stomachs, identified all contents down to lowest possible taxonomic ID, and then measured the blotted wet weight of each stomach content item. And so here's some examples of some of the stuff we found in the guts of these species. Here, on the left, we have from the stomach of a sea bream, some red drift algae. Here we have a Caulerpa rhizoid. On the right, we have some green filamentous algae taken from the stomach of a sheepshead. Looking more at these diets, on the left, we have some haliduli, which again is the seagrass we're going to be focusing on, as it's the main seagrass present in the Indian River Lagoon. And then on the right here, we have more haliduli, as well as a whole brittle star taken from the stomach of a sheepshead. So taking a preliminary look at the collected data, this graph shows proportion of gut content mass on the y-axis across the 5 most abundant items in the stomachs of these species on the x-axis. And so, and this graph has, sheepshead in red and sea bream in blue. And so what we can see from this graph is that red drift algae here contributes a lot to the diets of these species. And it's followed by unidentified material, which is not uncommon in gut content analysis, then by haliduli or seagrass, green filamentous algae, and then Caulerpa. And so we know based on this that both of these species do consume seagrass. However, it seems like it's in small amounts. But when we take a more holistic look at the diets of these species. Again, this is an NMDS. NMDS is an ordination method, and all that means is that each point represents one fish, and the closer 2 points are to each other, the more similar the diets of those species. The farther apart, the more different the diets of those species. And so when we look at this data cloud, we see a lot of overlap between sea brim in blue and sheep's head in red. So very high similarity. I also matched this analysis with a function in the OSTATS package in R that measures niche overlap. In this case, it's dietary niche. And we had a niche overlap of 0.934. The max for this statistic is 1. So we saw really high overlap in the diets of these species, suggesting that they could be competing for dietary resources in the Indian River Lagoon. However, it's really important to recognize that diets are very much influenced by resource availability in the environment. So if a resource is not present to be consumed, we will not see it in the guts of these species. So for example, if you have a sea brim in any given location in the Indian River Lagoon, and there's no seagrass around because a lot of it has died off, then we won't see seagrass in the gut of that sea brim because it's not available to be consumed. That doesn't mean if it were present, the sea brim wouldn't consume it. It just means it wasn't around for the sea brim to actually eat. So we need to consider that when we're actually trying to quantify the diets of these species. And to do that, we matched our gut content data with data from St. Johns River Water Management District's long-term seagrass monitoring data set. At each of these sites, twice a year in summer and winter, they measure the percent cover of seagrass and macroalgae in quadrats every 5 meters until the end of the seagrass bed. So you get estimates of percent cover of seagrass, macroalgae, and you also get transect length as a proxy for seagrass bed size, because they keep going until that seagrass bed ends. And so we paired these data with our gut content data based on seagrass site proximity and sample season. So for example, a sheep's head captured on the north side of Sebastian Inlet in the summer of 2020 would be paired with seagrass data from this site collected in the summer of 2020. And so to relate these two data sets, I used another ordination method. This is a distance-based redundancy analysis. It's similar to what we saw before. The closer 2 points are, the more similar the diets of these species. But the unique thing about this analysis is it allows us to look at the diets in the context of environmental variables. And in this case, it's our seagrass and macroalgae data. And so it also allows us to conduct model selection to determine what environmental variables were most important in determining the diets of these species. So when we did that model selection, we found that fish species, haliduli percent cover, transect length, which is again a proxy for seagrass bed size, and latitude were the most important variables driving the trends we see in these diets. And so when we look at the actual ordination, we see the clear influence of fish species here. Sea brim are grouped here, and sheepshead are grouped over here. And when we look at our continuous variables, which are displayed by these vectors here, they tell us that as we move from this point in this direction, that the percent cover of haliduli and the seagrass bed size in the environment around where these fish were collected increases as we go this direction. So in other words, these fish were captured in areas of higher haliduli percent cover and seagrass bed size, and these fish were captured in areas of lower haliduli cover and seagrass bed size. Latitude is likely a consequence of the distribution of seagrasses in the Indian River Lagoon. As we decrease in latitude, we tend to have bigger seagrass beds and higher percent cover of haliduli, just because of the tidal regimes present in the Indian River Lagoon and flushing of nutrients. And so what I did is I kind of grouped these samples into two groups. So this served as the high seagrass group, and this was the low seagrass abundance group. I then used a similarity percentage analysis to determine which gut content items contributed the most to the differences between the diets of high seagrass fish and low seagrass fish. And here we're looking at the results of that analysis for the low seagrass group. Here on the y-axis, we have proportion of gut content mass across the four most important items contributing to the differences between high seagrass fish and low seagrass fish. and it identified red drift algae, unidentified material, haliduli, and green filamentous algae as being most important to the differences between these groups. So this graph likely looks familiar because it's very similar to the graph we saw early on. It has high contribution of red drift algae with low contributions of all the other items, identified material, haliduli, green filamentous algae. And here I want to highlight red drift algae and haliduli, because I think that's really where the story emerges. Because when we look at high seagrass sites, we see a much lower contribution of red drift algae to the diets of both sea bream and sheepshead, and a much higher contribution of haliduli to the diets of sea bream. So in other words, when more seagrass is present in the environment, we see higher consumption of seagrass by sea bream. So to synthesize these results, one, we found that sea bream and sheepshead consume seagrass, and they also consume many things very similarly. Their diets heavily overlap, which means sea bream could potentially negatively impact sheepshead in the Indian River Lagoon. Third, sea bream consume seagrass, but do so even more in areas of high seagrass cover. So sea bream have the potential to, one, graze seagrass, two, graze them even more when they're in higher abundance, and could potentially impede recovery as they become more common in the future. And this could potentially negatively impact not just sheepshead, but also other important ecosystem components of the Indian River Lagoon that rely on seagrass, like manatees, fisheries that rely on seagrass for foraging grounds, etc. Finally, we saw that Caulerpa really wasn't that important to sea bream or sheepshead. We really didn't see that much Caulerpa in the guts of any of these species. And so when we take that understanding and apply it to what we know in Crystal Bay about seagrass and Caulerpa systems, we know that, or we can synthesize that seagrass and macroalgae do support unique communities in natural conditions. We also see that fauna respond to changes in seagrass and macroalgae abundances. And finally, we know that climate change, eutrophication, and other anthropogenic stressors may impact the communities living in seagrass and macroalgae, and may do so differentially, as we saw sea bream interacted with seagrass much more than it did macroalgae, Caulerpa. There's a lot of people to thank for this project: the Fisheries Independent Monitoring staff in Melbourne for helping us gather fish, Caitlin Tucker, Ari Siegel, Tegan Fraser, all the other people that helped me process all these samples. There's so many of them, I'm not going to sit here and list them all, but thank you to all of you, especially for helping me with these samples, as well as my funding sources as well. Thank you.
Thank you so much, Adam. That's really cool stuff. Folks, if you have questions, you can either type them into the chat, or you can unmute yourself and ask Adam your question directly.
Hey, Adam, this is Morgan. I had a question.
Yeah.
So like as your macrophyte communities kind of merged in the fall, like it got more mixed, did your faunal community look similar to like your mixed assemblages from the spring, or was it like an entirely kind of different community?
Yeah, so it suggests it was a different community, because when we look here, I guess I'll exit out. What's going on here? Oh my. It really was a different community. There was an influence of season. So if I can, my computer will cooperate, I'll go back to this. And so here we have, so this is the fall, and then they were homogeneous in spring. And our mixed community from fall is here in the red. So we see that there's differences between the different habitat types in fall because of the clustering, but we also see from this graph, and one of the reasons I think it's really cool, we can also see the seasonal differences because the spring samples cluster away from the fall samples. So we know that the spring communities are different from the fall, and so not necessarily equivalent to what the mixed sites were in the spring or in the fall.
Okay, gotcha. Thanks. Yeah, I was driving, so I couldn't look at the graph. So thank you for explaining that.
Yep.
Adam, I had a sort of follow-up question to that. I was wondering how your communities differ, if they're totally different organisms, or if these communities are different because they have similar organisms, but higher abundances of some. And the reason I ask this question is that it might have some implications for how what happens when plants go or when the plant community goes from seagrasses to macroalgae or back. As you say, one of your results is that they recover, like that if the plant community changes, the animal community covers really quickly. But what might happen, that's in a system where it appears that they're changing regularly. What happens if it's a system that shifts to macroalgae for a few years and we do a restoration and it comes back to seagrasses?
Yeah, so it's mostly differences in the abundance of the same organisms, at least from what I've seen from preliminary analysis. I'm not sure if that would be the case in a system where you have macroalgae, taking over and then potentially seagrass coming back. I imagine that again, this goes back to the macroalgae being the bad guy in a lot of from a lot of media sources, but I think macroalgae would actually provide some kind of refuge for the organisms that tend to live in seagrass rather than just total replacement. Though I guess under long-term you would definitely see changes long-term if macroalgae persisted and seagrass didn't recover.
I have a question about kind of just the kind of composition of the communities. But Charles has a question, so why don't you go ahead and take that one first?
Okay.
I can read it out.
Okay.
What is the size of the test area in Crystal Bay?
So it's this right here. It's not very far, or it's not very large range. And that's because the purpose of this study was not to necessarily sample all of Crystal Bay, but we wanted similar abiotic conditions. We wanted to really study differences between macroalgae and seagrass. So, we went about, when we were choosing these 11 sites, we went about 500 feet or so between each site. So my guess is a mile and a half in total, maybe a little bit more, but it's not far. But the nice thing that came out from that is that abiotically speaking, both depth, temperature, salinity, all these sites are almost identical, which makes it a really cool way to look at the differences contributed by macroalgae versus seagrass.
And Charles had a follow-up to that and asked if any sampling has been done north of Cedar Key.
North of Cedar Key, our sampling, no, I haven't. I'm not aware of people working on macroalgae versus seagrass up there that way either, but I've heard people talk about Caulerpa doing well, I'll say, up that way. But that's all I know.
There's another question in the chat from Robin, and they ask, does Thalassia testinum slough off most of their leaves in Crystal Bay during the fall? If so, was this considered in the spring samples? Was sampling done prior to the spring growth of new blades?
Yeah, so that's a good question. So here, for ease of communication, we call it fall and spring, which it was in the fall and spring. But we looked at this at the beginning, or at the fall being the end of seagrass growing season, when we in theory had maximum seagrass standing crop, and then we did it at the beginning of the seagrass growing season before really we saw major growth of seagrasses. And so I think that's what's contributing to the differences we're seeing, is we see that senescence of seagrass above ground biomass over winter, and then when we sample again, they really haven't had time to grow and establish. So that's why we see a kind of a more homogeneous mixture. You have some seagrass blades, you have some macroalgae across almost all sites, whereas as you move in fall, seagrass kind of have the ability to come to dominance, I guess, or grow in, and so you have habitats that are more just seagrass or more mixed. Did that answer the question?
I think so. You're getting the thumbs up.
Okay.
Sorry, Savannah. There's more questions coming in the chat. Lauren asks, remind me if the FIM data extends into Mosquito Lagoon. We have quite a lot of seagrass still remaining throughout Mosquito, so I'm curious if that could even enhance that poleward expansion due to higher seagrass availability.
Yeah, for the Seabrim, yeah. So there is FIM data in Mosquito Lagoon, but they only do it probably every few years whenever they get funding for it. So for example, when I interned in the FIM lab in 2016, they had some federal money to go sample there for a year or two. And then when I was hired there during my gap year, when I started this work, which was 2020, they had just got another grant. So I actually helped run the Mosquito Lagoon stuff that it started after I got there. So it's sparse data. But if you wanted to do some seagrass FIM data relations, it probably could be done. So the temperature of the water also is really strongly related to the rise of Seabrim in the Indian River Lagoon. They've seen, I think our area of the world is predicted we should have seen a 1 degree Celsius rise in sea surface temperature at this current time. And that's exactly what we see in the lagoon. We see a 0.9 degree Celsius increase in Indian River Lagoon water temperature over the past 30, 40, whatever years. And so that's when we did the PCAs and the different, FIM has their standard GLIMIX mixed modeling approaches. You really see that temperature contributes to Seabrim increases. And so I think the temperature isn't quite there in Mosquito Lagoon yet for them to enter. But once I imagine that once the temperature is there, or maybe it already is and the Seabrim just haven't got there, I would expect we'd see very quickly an increase in Seabrim in Mosquito Lagoon. Mosquito Lagoon, because it's the southern part of Mosquito Lagoon that has most of the seagrass, is my understanding, but oceanographically speaking, my understanding is that Mosquito Lagoon is very well connected. So I think Seabrim would have no problem going up the coast and entering Ponce Inlet and going back down there. I think they might be a little limited moving upwards through the interior of the lagoon by salinity, but we'll see. That was a little more of a complex answer than needed. Got on a tangent there.
Justin asked if the IRL study of spirit diets is published.
Not yet. Hopefully we'll be submitting this fall. It's ready to be written up.
He follows up by saying in 2017 to 2019 seagrass die off in the IRL, red drift macroalgae, especially gracilaria, is a dominant macro by biomass. Mm-hmm. Are there any predictive models available that show a shift in Seabrim abundance?
Shift in Seabrim abundance based on red drift algae?
I think so, yeah.
Biomass? Those models have not been constructed that way. I'm not sure if red drift algae is necessarily contributing to the rise of Seabrim, or if it's just, that's the most abundant by biomass, and that's why we see it so abundantly in the guts. That latter one would be my guess, because despite seagrass percent or haliduli percent cover and drift algae percent cover being positively correlated with each other, when there was more seagrass, we saw less red drift algae in the guts and more seagrass. So my guess is there wouldn't be a huge driving force by red drift algae on Seabrim abundance and Seabrim rise in the IRL.
I think we have a gap in the chat. So Savannah, if you want to hop in.
Yeah, so back to the Nature Coast region for your communities, did you first question is about the functional identity, so like grazers, like filter feeders, like were your assemblages dominated by grazers? And then the second question is, within the grazers, did you have mostly like crustacean dominated grazer communities or gastropod dominated, and did those flip when you saw increasing dominance by seagrass versus macroalgae?
Yeah, so I'm not sure. I haven't done like actual analyses on the actual functional groups themselves or based on taxonomy or anything like that. But when I look at the simper on individual species, it seems like the Caulerpa paspaloides tends to have a lot more gastropod mesograzers, whereas the mixed Thalassia Caulerpa tends to have more crustaceans. And then by the time you're at alt Thalassia, you have crustaceans and more fishes like Lucania parva, etc. Pinfish, I'm not sure if there was a relationship there. 'Cause I consider them kind of, I guess they're a grazer, mesograzer, and predator.
Thanks. Yeah, I was just wondering, because there's some work that suggests that the gastropods might be less available to like upper trophic levels, a little bit harder to prey on. And the crustaceans might kind of enhance that secondary production. So that was why I was interested in that.
Yeah, I'm not sure. I mean, as you know from our committee meetings, I think some meta community analyses based on functional groups can help get at that.
I have a follow-up to that, just really quick. Are there organisms who might prefer Caulerpa? Like you basically, you showed in the IRL that your fish might eat some, but they don't prefer it. Do you think that there are organisms that might thrive on diets of Caulerpa?
Yeah, I think so. I definitely think that there's a lot of similar, like shared species between them, but definitely, across many ecosystems, we see increased diversity and abundance of a lot of invertebrates in Caulerpa habitats compared to seagrasses. Mark Hayes' work suggests that more immobile species would be better off, they would be better at thriving in Caulerpa habitats because immobile species have adapted to tolerate the toxic chemicals in Caulerpa, whereas animals that can move, like parrotfish, whatever, in theory would prefer to chew on seagrasses, which the secondary metabolites in theory are not as toxic. At least that's what Hayes experiments suggest. There's a documented case of Sarpa salpa, which is a sparid in the Mediterranean that actually really likes to consume Caulerpa over there. So I think there's definitely some specialization that some animals do prefer Caulerpa and some animals prefer seagrasses, and that's why both are important, because a mixture of everything can support a more diverse, complex assemblage.
Great, thank you. Anybody else have any follow-up questions for Adam? All right, if not, I think we'll go ahead and end this. Remember, we do have one more seminar. It'll be in next month, and before we take a break for the summer. So I hope to see y'all back next month. Thanks again, Adam.